The EEG, or electroencephalogram, is a device that measures electrical activity in the form of action potentials and nerve impulses transmitted along neurons. These neurons are close to the scalp and can be picked up by the EEG, although it cannot read deeper brain electrical activity. These action potentials are measured by electrodes placed on the scalp. The potential areas for events were at F3, F4, P7, and P8. The device that was used was the eMotive Epoch X EEG headgear with 14 channels. The eMotive kit came with several things, including felt tips with saline, a USB Bluetooth receiver, a USB Type-C cable, and the device was sampled at 128 Hz. During the setup phase, the participant would be asked to sit in front of dual monitors at an i9 mid-tower desktop and place the EEG device on their scalp. The EEG device was then adjusted by the researchers, and saline was added to the felt tips to ensure good contact and EEG signal quality. During this time, the EEG device was turned on, and the eMotive software would begin as well. The contact quality between the USB receiver and the EEG device was ensured, and this began the setup phase to ensure good EEG quality and good contact quality. The EEG band would be attached to the top or back of the head, and all of the EEG electrodes would be placed onto the scalp in accordance to the 1020 electrode placement. After all of this had been verified and ensured, the process of recording EEG signal data would begin. During this time, the EEG signal could be monitored using the tab to the top left. This would show all of the electrodes with good connection quality and the signals in accordance to time. As is observed, all 14 electrodes have signal quality for each electrode in accordance to the 1020 electrode placement and the 14 channels of the epoch. During this, you can apply markers to the beginning and ending of each trial. This was an example of how those markers would be placed. When beginning to record, a test name would be picked out, as well as the subject name. This is a demonstration of how that process would be started. In addition to the subject name and the title of the trial, a baseline would be selected, which gave a distinction between eyes open and eyes closed. This could be used for comparative data of other events in the future. This is an example of the first 15 seconds of the baseline, where the participant would be asked to sit with their eyes open and relaxed and not move for 15 seconds. After these 15 seconds are concluded, an additional 15 seconds would be asked of the participant to sit still and quietly for an additional 15 seconds. During this time, this baseline would be able to distinguish between eyes open and eyes closed, and things that could be observed from this are fluctuations between the eyes open and eyes closed. These could be known as artifacts, or anything in addition to the things that do not need to be observed. During this, you can see a demo of a test where the participant was asked to interact with the 3D cube. This was then monitored by the EEG eMotive Pro software and would be a demonstration of how one of the tests would be run during the experimentation. Following this test, there is a brief demo of how markers would be placed beginning and at the end of each trial. These markers are apparent in the eMotive software and also stored as exportable data that could be matched up with EEG data outside of the eMotive epoch and its component software. The EEG recording would then be stopped, and this additional information would be stored as well. Other things that can be observed by the eMotive Pro software is the distinction of different signals from the 5 to 40 Hertz range. However, for this experimentation, the only range that was looked at is the 12 to 25 Hertz beta frequencies. Different electrodes can be viewed at the same time, such as the A, F3, and F3 locations eMotive software. During this time, you could also see other factions such as excitement and focus in the eMotive Pro software as well. During this, you can see an example of the Ericsson flanker task that would be used to gauge and configure the eMotive EEG headset and also view EEG signals during the entire process. This could be used in the future for other attention-related tasks, but was only used as a foundation for this research. After each trial was done, the files could be exported from the eMotive Pro software. This eMotive Pro software allowed for the files to be exported as a CSV file or EDF file. For our purposes, we exported them as an EDF file. These EDF files were then imported into MATLAB into a program called EEG Lab. EEG Lab would be used to analyze the signals, to filter, and also re-reference the signals. This allowed for clean signals and to discard any artifacts of any information that did not need to be observed. At this time, this is a representation of a cleaned and filtered EEG signal and only 5 electrodes specifically. After viewing this EEG signal and verifying that everything was correct, this signal could then be processed into another form that could be viewed and observed by observers and the researchers in a very easy to manage way. This would put them inside of a spectrum map that was filtered from a range of 5 to 35 Hertz and placed on particular events. During this time, you can see activity in accordance to color. Any time that you would view a darker color, such as blue or baby blue, this would indicate 0 signal. Anytime you'd see green, you would see 0 signal. Anytime you'd see yellow, yellow would indicate about 5 volts or microvolts of signal. And anytime you see anything above the yellow, that is a higher microvolts frequency that is given from the EEG. This allows to see certain amounts of power at certain frequencies that would correspond with the waves, such as the beta waves, the alpha waves, the delta waves, or gamma waves. Thus, this concludes the demo of the research portion of the project.